Okay. It's Friday, the 21st of May, 2010, and Horace Barlow is being interviewed for the Physiological Society Oral History Programme. And and that's Horace, David Tolhurst, Anne Silver, and Martin Rosenberg, are in the small meeting room in the Cambridge Physiological Laboratory. So, first of all, Horace, thank you for agreeing to take part in that. And before we get on to your career in physiology, we'd like to hear a bit about your family and where you grew up and your schooling, your early interests, and particularly the Darwin connection. So, far away.
Well, I was born with a scientific silver spoon in my mouth, so to speak. Both my parents both of my grandparents were scientists of their day. On my father's side, it was Thomas Barlow, who was physician to Queen Victoria and was a genuine pioneer in applying real science to medicine of that time. A very well-known figure of that time. In fact, he even had a disease named after him. Well, it's a disease named after him in Germany, but not in England.
What was the disease?
Das Barlowische Krankheit is scurvy rickets. Is rickets due to scurvy rather than vitamin D deficiency? He differentiated the two. And he lived to a great age of 99 3/4 or something. But my father, his son, was not a scientist at all. He read classics at Oxford and became a senior civil servant and was a wise old bird who knew how to get round corners everywhere, like that character on the radio. What's he called? Anyway, but on my mother's side, my mother was a Darwin, grandchild of Charles, and very keenly interested in both in Charles Darwin himself and in Darwinism all her life. And she was, like most females of that age, never went to university, but she did do quite a lot of scientific work with William Bateson, who was the rediscoverer of Mendel's work. And she was a very good botanist and knew the names of all the plants and tried to teach me without any luck whatever, But she did teach me about Darwinism, which was, it was obviously always in her mind, and she would point things out all the time. And so I think I learned a lot from her, actually. She would point out interesting little things, all the time. And then that was my first stroke of luck. The other one was being sent to Winchester, which was a school where the scientific teaching was very good. And that meant that it had a lot of very good scientists there. So I learnt what a lot of people only learn when they come up to university, that they're not quite the top of the barrel, so to speak, in the things they think they're good at. Because I was in the same, sort of more or less the same vintage as Freeman Dyson and James Lighthill and Mike Swan, and there were several others who I very quickly learned far outshone me in my mathematical and physical abilities, so to speak. So I thought seriously of going into biology. But then the war came along, and I came up here, in fact, I think 2 terms earlier than I would normally, and as a medical student. And I first of all took the remaining first MBs, which I hadn't taken, which were one in biology and one in organic chemistry, I think it was. I think the organic chemistry exam was the only one I failed ever. In the biology one, I didn't learn much that I didn't know already, except I saw the insides of a dogfish, etc. So, what more do you want in that line?
Yes, and so that had got you to just about the beginning of the war.
Yes.
And then what happened after that? I mean, you left school.
Yes, I came up here actually in January 1940 and studied for the remaining parts of the first MB, which I hadn't got, and then started the year, so to speak, in September 1940, October 1940.
So were you weren't too young to be involved in any the war?
No, well, I was a medical student. And
that was reserved.
Yeah. And so I did the shortened medical course here then. But then I spent a year, just at the end of that, I'd just finished Part 2 physiology, and I spent, I don't know, it was probably 2 or 3 weeks, perhaps a bit longer than that, with William Rushton. And he showed me how to get action potentials from the giant nerve fibre in the earthworm. And I asked him one day, well, why do we have to do all this dissection? It's a very big nerve fibre. Can't we just record it from the whole worm? And he said, that's a good idea. We'll see if we can do it. And we did it, and we published something in Nature. So that was my first publication.
So that was as an undergraduate?
Yes. Well, just finished the course, yes.
Do you think you impressed the people here? Did they say, wow, this is somebody who will be meteoric, or...?
I don't think enormously. I mean, they were used to the likes of Hodgkin and Huxley, don't forget.
Who were your tutors at that time?
Well, the ones I remember who supervised me in physiology is actually I didn't want to be supervised by William Rushton because I thought his way of thinking was too much like mine, and that I would learn more in things I didn't know much about from others. So I did have one supervision, I remember, from Rowton. Then he went off to Harvard, and another from somebody called Parsons, I think it was, who did some Trinity supervisions.
Dennis Parsons, I think.
That may have been. He smoked a pipe. That's all I remember about him. But then I went with Feldberg, which was great. That was a subject which I sort of wasn't very good at, and he was here
then.
Yes, yes. And then I don't think I actually had a supervision with William, but I spent many hours talking to him, of course. And
Can I just ask you one thing, which I think ought to be recorded if your answer is yes? You mentioned Rowton. Now, did he, at that stage, feed his handkerchief into his mouth and feed it out again?
Yes, he did. I never saw him do it, but my brother Andrew did. And because he went to see him about change, he was an engineer and he wanted to change to medicine, so he had to go and see Rowton, and he went to see Rowton, and he did this. And when he got the handkerchief out, my brother Andrew turned to him and said, why do you put your handkerchief in your mouth, if I may ask? And he said, I find it helps me to think.
Comes out the same colour, does it?
I wasn't there.
When you'd finished your degree here, your undergraduate degree, you then went to Harvard. Did you go directly to Harvard, or was that when you went to Mill Hill?
No, I went to Mill Hill. Well, it wasn't Mill Hill then. It was Mount Vernon. It was which then later became Mill Hill, so to speak, when the MRC built a grand and new establishment.
Was it in the same part of London?
Yeah, well, it's in Hampstead actually,
yes.
And it was it was our main intramural laboratory, so to speak, and had a lot of work going on. And G.L. Brown had a unit there doing naval personnel research, which was chiefly concerned with diving, which I didn't know anything about, of course, but it was just sometime before D-Day actually, but they were concerned with harbour clearance and one-man submarines and that kind of thing. So it's improving diving gear, which they didn't really have any ordinary what every holidaymaker has now, and we have
diving.
So we had to, and the chief problem that they're working on was oxygen poisoning, actually, which is what happens if you, I didn't know anything about it and haven't even heard of it, but if you breathe oxygen under more than 1 atmosphere pressure, and particularly if you're in a diving suit, and I don't know if anyone's ever still discovered why it's so bad to be wearing a diving suit, but you get convulsions and things like that. And so they were doing experiments on that.
Was this with big helmets and lead boots?
Well, no, it actually had a compression chamber.
Oh.
But we thought that some cases were misdiagnosed as oxygen poisoning and were actually caused by carbon dioxide accumulation, because the diving gear they had, shallow water diving gear they used, was entirely the Davy submarine escape apparatus, which had a small canister for absorbing CO2, and we reckoned that when people were working hard, it just over.
So who else was at Mill Hill then?
Mill
was
Hill.
Harold Lewis there?
Who?
Harold Lewis.
No.
Who was the temperature regulation?
No, the person he was into the thyroid. What was his name? He was the boss of the place. But there were a lot of interesting characters there too. There was a man who tested Winston Churchill's cigars, and he said it was a very good job.
Wasn't Heinz Wolff there then?
No, he wasn't. I knew Heinz Wolff a little bit. He was a friend of my older brother's.
How long were you there?
Only, I think it was just about a full year.
And then you went to Harvard.
Yes, this was a Rockefeller medical studentship, and I think it was a total of 20 of us, about 6 for 3 years or so, went over there. And
what year would that have been?
that would have been starting 1941.
Oh, it's still
No,
wartime.
44.
Yes, 244, yes. And that was interesting too, because of course by that time I was quite seriously interested in physiology as a career rather than medicine as a career.
How did you get to Harvard in wartime?
By boat.
It's quite tricky.
Well, it was. Quite a lot of people were torpedoed on the way. The mortality rate in that 20 was astonishing. It was, I think, 2 people were torpedoed, another 2 got TB, and it was.
And did going across, did you take a sort of a zigzaggy course
Yes,
and everything?
Yeah. And not in a convoy, because it was on the Queen Mary, and Queen Mary went fast, and they reckoned that it had to be a pretty nippy submarine in just the right place to be able to.
What did your parents think about this?
Sorry?
What did your parents think about this?
Well, I think they were quite glad to see me. England wasn't a very good, very low-risk place to be at that time. You know.
America just gone. Was it 41 they joined?
42, yes.
42.
Did you do any lab work or see anybody's labs when you were there? Or were you...
Yes. That was the closest I got to ever actually being a doctor, because they had the policy of putting third year and fourth year medical students, giving them temporary jobs looking after a ward over the night, you know. Of course, that was a terrifying experience, but it's quite instructive. But the other aspect of American medicine was that it's very much more science-based than English ordinary practice was. I mean, I think the research labs in England were every bit as good, but the actual ordinary doctor always did tests almost before they'd done a physical examination or taken a history, you know.
And then when you'd finished that, you then came back to Britain again, and the war had finished.
Yes.
And
yes.
you had a post at, was it University College?
I had to do. The Harvard degree didn't actually enable me to practice even in America. Certainly not in England. So I had to do another. I think it was 18 months of clinical training here to get the appropriate places in the grey book signed, and
Where did you do that?
at UCH.
At UC. Mm-hmm.
And that's there you work with Walsh and Cohn?
No, that was actually when I was at Harvard Medical School.
Oh.
That was Henry Cohn had heard a story about people carrying degaussing coils on their shoulder, which had current flowing through them, and had seen flashes. And so we decided to take that up, and quickly discovered this had been discovered about 5 times before. But we assumed that it was probably the induced current that stimulated, because the eye is very sensitive to currents.
You think it was the eye rather than the visual
Yes.
cortex? You can do transmagnetic stimulation.
Yeah, you can do that, but you need very high fields for that.
So while you were officially doing your medical studies, you get 2 papers in the American Journal of Physiology.
Yes, that's right. Yeah.
First author. Was that because...
Alphabetical.
Is it alphabetical, or who knows?
I think that both Walsh and I were English physiologists, and therefore agreed to alphabetical order. And Henry Cohn, yes, I'm a bit surprised he agreed to that, because he ought to have been really, since it was his problem and we were just helping out.
Did that was 2 papers on different aspects of vision. Did that make you think that you'd like to do vision science, or?
No, vision had been one of the things I've been interested in the Part 2 course. So I've been, so to speak, a specialist.
Anything in particular that triggered you in the Part 2 course?
Well, I gave a talk to what's called the Natural Sciences Club when I was an undergraduate, and I think I gave that on I think I gave 2 actually, one on colour vision and one on absolute thresholds. That was an undergraduate society, and they had and we took it in turns to give talks. So I was already quite keenly interested in that. And then I had the choice when I was qualified either of entering the serious rat race in medicine, which would involve house jobs, residences, and so on and so forth for the foreseeable future, as far as I can make out, or trying to do science. And I thought I'd have a go at that first. So I didn't do any more medicine.
And then you came back here and sought out Lord Adrian.
Yes, with some difficulty. Yeah. He was reckoned to be quite a difficult fish to catch,
Well, was he still very active, or had he... He'd already got his Nobel Prize by then.
Oh yes. Oh, he was very active on 101 scientific committees.
He had about 6 years left, if I remember, of head of this department. In 47, I think. Did he finish in 53?
That sounds right. Yes.
But you didn't go to him to, in a sense, do his kind of research.
No, he was a very hands-off supervisor.
Oh, was he?
And he saw me for 5 minutes and said that he thought he could get me a research studentship, and that was that. And then we talked a little about things that I might do. He knew I was interested in vision, and he said that he'd read an interesting paper by Marshall and Talbot on the role of microsaccades in keeping vision going, so to speak. Was it really true that if the eye was completely still, you would see nothing? And so I spent the first year doing that, and decided I had to find some equipment. So that faced me with... Well, by that time I was not looking for Adrian, but for Adrian's assistant, Leslie.
Hatton.
Yeah, Hatton, that's right. Yes. Who was almost as difficult to find as Adrian, actually. But eventually he showed me what recording cameras were available. And of course there's nothing remotely suitable for recording eye movements, but there was one very old movie camera that had the mechanism for moving the film on, at any rate. And I reckoned that with a little bit of
The fascination then was with single fibre.
That was later.
That was and Zoterman.
Yeah.
And granite as well, yeah.
No, I didn't work with either. I knew them, of course, both them.
Was Lord Adrian's assistant technical staff? Was he a batman, basically?
Yes.
I think so. Probably lit the fire in the lab in the morning.
Probably, yes.
Boiled the kettle.
Yes. And kept visitors away.
Yeah. I had a peep this afternoon at your paper on eye movements. It's fascinating for all sorts of reasons. But one thing was the Journal of Physiology had, 1,500 pages that year. I was surprised how many.
Yeah.
There were 3 volumes of about 500 pages each, and I think last year we had one of 6,000 pages. But the other thing was there weren't that many journals about, and there wasn't much literature. Your papers in those days had about 10 references on which, and presumably that was a thorough reflection of the scientific knowledge at the time.
Well, that would depend upon how good a job I did. If one did a really thorough search of literature in those days, of course, you had to be quite fluent in German, because a large part of the physiological literature was in German.
Well, Yarbus, was it Yarbus was Russian?
Yes.
And what sort of date did he publish his pictures of where people looked at, while they were looking at pictures of people, I think it was?
The pictures of pictures they were, actually.
Yes.
It was one of the Soldier's Return. Do you
Oh,
remember?
yes.
And there was another one of the subject looking at a portrait of his girlfriend. I got into trouble with that because there was a visit from the Russian delegation, and everything had to be translated, of course, even though some of the Russian delegation spoke quite good English. And I asked him to ask Yarbus whether the subject had her clothes on or not. Because
you thought that might change where the person would look.
Yes.
Yeah.
And where the results showed that it could fit either the face or not, you know. And Yarbus immediately picked this up and gave a little snicker and said it was how she had her clothes on, you know. But before he could really get that answer in, the interpreter had said, I refuse to translate that question.
Mike Lander's done some work recently on.
Oh yes.
Have you seen that one?
Oh yes. That's very nice. Have people playing ping pong, making a cup of tea, that kind of thing.
Learning music.
Yeah,
First time they look at every note, and then as they learn the piece more.
Yeah. Well, an experienced sight reader doesn't read every note. They go.
But you were right at the beginning of that interest. Yeah.
Did you ever publish another paper after that one on eye movement?
No.
No, it was the only the one. Yes. Would you like to tell us how you measured the eye movements? Because I don't think you'd get permission to do that now.
No. I looked into the sort of optics and mechanics and so on, and people were busy using reflection from the cornea. But if you work out where the image is when you're looking at a reflection of a bright light in the cornea, it's way back inside the eye. It's only 2 or 3 millimeters in front of the centre of rotation. So it's clearly, it's very insensitive and easily disturbed by head movements, which you have to get rid of, otherwise you confuse them with eye movements. So I thought it'd be at least 3 times better if one put it a bright spot on the surface of the cornea. So I put a little mercury droplet there.
Yes. He all sounded terribly uncomfortable. You talked about these wedges and white bars.
Well, that was to keep the head movements down, because the head movements completely ruin it. So
and then you said that
it was a very convenient. It had been used by. I was in the room that used to be. There was one where Rowton did his blood flow measurements, his rapid reaction things, and it had a concrete slab let into the wall for balance, as a stable thing, and that was very useful for putting the head on. And then there was I think it was an old microtome or something like that, which had an iron frame that I could attach to the thing and put a wedge down one side and head on the other. That kept their heads pretty steady.
Who were the subjects?
Just friends and other PhD students.
The system where you have a contact lens and like on a stalk and a mirror, was that yours?
No, that was what Jarvis had. Was a contact lens which was sucked onto the eye, and in addition had a stalk and a magnifier and a lens, so which focused on what was on the stalk. So you put a fixed image there, and with various, you could do other things with it too. And it was Briggs and Ratcliffe who did the method with a mirror.
Yes, to stabilise.
Yeah.
So you're good. You're good with your hands. You like making things as well as.
Yes. Of course, you don't do that in physiology now.
No. But you.
I'm sure we would if we could. But we don't have.
You know your way around a workshop and
yes.
lathe and things.
I was reading Dave Hubel's memoirs about his early life in research, and he was obviously an absolute maniac for making things. And always throughout his career had a metalworking lathe in his lab, and went on making gadgets all through.
I remember seeing Michael Daly's father in Buck and Ryan's with a pleasure on his face when he was looking at these tools.
Yeah.
So by sort of the end of sort of 48-ish, perhaps, you've done worms, you've done electrical and magnetic stimulation of human eyes, you've done eye movements, and somewhere you thought, let's look at frog action.
Well, I came to the conclusion that eye movements weren't going to get very far. I talked to Adrian. He continued to be very. Every now and then he put his head round the door and say, "Have you read such and such a paper?" And we'd chat for. I would estimate not longer than half a minute at most, but they were very beneficial half minutes. And then on one occasion I said I thought I'd come to the end of the eye movement stuff, and I wanted to do some recordings from Frog's Retina to repeat some of Hartline's work, at which he said, "Oh, I wouldn't do that. Hartline's a very clever chap, you know." But I didn't have time to explain why I wanted to do it.
That was Stimulus, wasn't it? Hartline?
Well, he'd earlier recorded from frogs.
Right.
Yeah.
You did that with I suppose we'd call it an isolated eye cup now,
Yes,
wouldn't we?
yes.
Did you perfuse it with anything, or did it just sit in the air
It
and
happened
dry up?
to dry up.
Yes.
I tried to keep it from drying up, but I found as soon as we put bringer's fluid on it, it killed it, so I stopped doing that. But eventually, with Adrian, he hadn't said anything about it except I wouldn't do it. So I thought I'd better write out why I wanted to do it. I did that and sent him a little note saying that I was planning to go and get some equipment which I'd always bought from the leftover government stores, and got power supplies, and those who were in the know knew what came out of which plugs and so on and so forth.
Tottenham Court Road.
Yes.
Or Lyle Street.
Lyle Street.
Yeah, I used to go there, and people used to wonder why.
Yeah. And you got stuff there at sixpence a pound. You know, whatever you took off the shelves, they weighed it, filled it with sixpence a pound. So I got to. One of the ways in which I used to make noise was to put a photon multiplier inside a black box with a light, and with a DC light, and record the output, which is very noisy, which was photon noise. That was as near as I could get to white noise.
That seems to anticipate a page and a half of your CV, doesn't it?
Yes, quite.
You prefer to work on cold-blooded animals rather than move to mammals, or maybe you didn't?
Well, I worked later on mammals, but I started off on frogs. because it's simpler.
Simpler.
When you were presumably eventually writing up, the paper was published in 1953, so that was probably quite a long time after you did it.
Yes.
Were you aware of Stephen Kuffler's work on the cat at the same time? Because your papers are seminal in this, aren't they? Both in 1953.
Yeah, actually my first paper was before, on this was, well, it's not proper paper, but it was before Kuffler did his, which was...
International Congress.
Yes. Is that down there? No.
Yes, it was. It's on my list in 1950.
Yes, that's right.
Yes.
And I think that was before his first one, which was 1952. And he was actually at that meeting.
Oh,
He told me afterwards he hadn't come to my talk, but he had been at the meeting.
Yes, no comment. But your interpretation, the way that you wrote the discussion of your paper, sort of introduced this idea that maybe the receptive fields were actually designed for particular behavioural tasks, like seeing flies at however many inches, I think.
Yes. Yes, that was new, and that was of course because I was very much taken up by the sort of Lorentz Tinbergen approach to behaviour.
Did you have any antagonism, or were your ideas accepted straight away? Did you have any problems?
I think people were interested, but when you publish a paper in the Journal of Physiology, my experience is that your postbox doesn't actually overflow.
Well, there weren't many other journals. This was something I was wondering whether you can have a picture now of how many journals there were and how many neuroscientists or vision scientists there were in the known universe at that time. It can't have been
if
very
you
large numbers.
No. I remember at the International Congress in Denmark in 1950. But I think that the talk that I gave there, which was just a brief talk, there must have been 20 or 30 people there, and probably I could recognise a good 1/3 of them, even though, of course, it was international. And actually, I met quite a lot of people there for the first time. But I didn't meet Kuffler.
Has anybody worked on frog retina since? Do they still find the same things as you, or are they interpreted in different ways now?
Letvin, what frog's eye tells the frog bit's brain. They didn't actually record from the retina; they recorded from the colliculus.
Right.
And a few people have repeated that and done it on other animals and so on. But I don't think anybody...
A lot on the formation of visual fields in the cortex, Vicky Sterling, I think, at Mill Hill. But that's more recent years.
The projection.
The projection, yes.
They did gaze.
Gaze, yes.
Yes. No, they've done that, yes.
Do you still think, presumably they were off cells, but that those cells are fly detectors? Do you think they really are a specific piece of frog anatomy linked up to its tongue somehow?
Well, the trouble is that the on-off ones, which I think are most like fly detectors, they're not very different from the cat retinal ganglion, cat Y-type receptor. And that's always worried me. If you look at different species of frog, there are quite noticeable differences between the different species of frog. And I could sort of contemplate at one moment a lifetime of recording from frogs' toes, etc., and becoming world's expert on this. But I didn't do. I got interested in something else for some reason.
Yeah.
So how did you, or was it subsequent to the frog work or in parallel, that you started thinking about retinal noise and Purkinje shifts and
that was
thresholds?
That was continuing interest I already had. and also because of my interest with the information theory people, realization that noise in all sensitive instruments is always a problem. Surely it must be in vision too. So I was interested in that, and in why, for example, you didn't get the results you expect if you just measure the threshold as a function of background intensity. You would expect there to be a region where you get no change because it's entirely dominated by some internal noise. Then a point where the external noise comes in, where it'll go up linearly as it goes the square root of the background intensity, because that's how quantum fluctuations go up. But you don't get that. In most cases, you find a long region where it goes up as a square root, which of course fits the idea that that it is actually that normal vision is quantum fluctuation limited.
Yeah.
So I was interested in plugging that idea, and then I came across a paper pointing out that how very rapidly the expected rate of thermal degradation of a pigment molecule goes up when you lower the activation energy. Only a slight lowering of the activation energy, corresponding to a shift of the peak absorption from 500 to 560, is enough to send the thermal degradation rate up by an enormous factor. So I argued that this was what, in fact, why the high sensitive pigments were shifted to the blue, because it decreased.
Do frogs have red rods, or is it not as red as I think, and they're cold-blooded anyway? So.
Have you ever seen a rod? Because they actually look red anyway.
Oh. I thought there was some animal that had two kinds of rods.
That's because they absorb short wavelength light. So they're shifted to the long wavelength side.
So when were you say that you were interested in the information theory people, is that Shannon and Weaver?
Yeah.
And so on. Were they sort of publishing in the early 50s? And
Shannon's work was coming to light just at the end of the war.
And a lot of people in this department, of course, have been doing electronics
yes,
stuff in the war,
yes,
so they probably knew a lot about
yes, they did, yes,
electrical
yes,
noise and electronic noise.
Yes, particularly when recording small voltages, you need to know about it because your electrode's limited by noise too.
And radar. And
yeah.
Were you involved with Pirenne at all?
Yes, well, he came and worked with William Rushton, so I got to know him then. And we talked about a lot of these things. And I was very much influenced by his little book, Vision and the Eye.
Yes, I have that. Yes.
It was a very good little book. You know.
He had beautiful handwriting. He wrote me a note once. He designed an eye bath for demonstrating to students, a big thing with big lenses so you could show the errors of refraction and so on.
Oh, right.
And he sent me the diagram of this, and he also sent me a stereoscopic picture of it, and he also sent me a stereoscope, a mahogany stereoscope.
Oh, that's great.
A beautiful little note.
So you were doing this noise stuff in Cambridge, and then you went to Stephen Cutler's lab for a year. Was that so that he could learn what you were doing, or you could learn what he was doing, or you thought that
he was very untheoretical? He didn't understand it well, and said, well, and if he didn't understand it, he wasn't going to make comments about it or of either kind. So his eyes just glazed over, you know, and that was that. So it was no good me trying to tell him what I was interested in, you know, because he. But that was. I had used cats actually when I was at Mount Vernon. We'd done. I can't remember quite why we he had to use decerebrate cats. I think J.L. Brown had some experiments going on to see whether the spinal cord was also sensitive to oxygen poisoning or something like that. So that's where I learned how to do that. I don't believe that Kuffler knew how to do it, so I think he probably learned how to decerebrate a cat from me.
Who else was in Kuffler's lab when you were there?
It was before Hubel and Wiesel were there. They went there just after I left. Dick Fitzhugh was a person I collaborated with. He was another theoretician. It must have been real hard on Steve, I think. And he was sort of into Hodgkin Huxley stuff, equations. And he and I, we worked together on the cats on the absolute threshold, and how sensitive it was, and spent hours measuring up photographic records and so on. Before the days of computers, you had to do it all by hand, and doing the actual experiments.
You felt you'd achieved something, though, didn't you? When
Yeah.
you did it that way.
Anyway, no one will repeat this, said to himself.
Do you think, how would the experiments that you did in those days, would they have been how different would they have been if you had computers to control things and count things and so on?
Well, the big change, of course, is when people started using awake behaving animals, because one could look through the anaesthetics and see what one thought had those that had the least effect on on sensitivity. We were interested in sensitivity, and I remember that the barbiturates, which had the effect of more or less suppressing all the spontaneous activity in the retina when you got up to an anaesthetic level, but they were some of the most sensitive results we had. But I think, I can't remember what we finally used. I think chlorelase and urethane or something. Because urethane was a bit...
Well, that suppresses everything, doesn't it, urethane?
No, you get quite a lively, liveliness is judged by pinching its tail.
Yeah. But was it the controversy about nitrous oxide?
Nitrous oxide, yes. Well, that was
Blakemore.
someone wondered whether it was anaesthetic at all.
Yes, the Physiological Society published a sort of guidance on that, specifically, because it was queried so often.
Yes, I think they decided that it wasn't an anaesthetic by itself. It needed to be.
No. I think most people who were using it,
and so on.
especially if one was using it in conjunction with a Curare or anything like that.
They misinterpreted the laughter, I think.
The what?
Misinterpreted the laughter.
So, you've done frogs, you've done a year in with Kuffler looking at receptive fields in the dark and absolute thresholds and so on. And then somehow this led, or was this just another line, to the sort of 1961-ish papers that so many people now quote in the conferences about redundancy and coding principles and things.
That was from my contacts with the group in London, really. It was called the Ratio Club, and we met, I think, 2 or 3 times a term for a number of years, 4 or 5 years. There's been a book written about it by somebody in Sussex, and then it sort of petered out. But that I talked about redundancy there, and learned a lot. There was Donald Mackay was in it, and a computer scientist from Malvern, Philip Woodward, and he was all. They were. Donald Mackay gave excellent talks, and I only discovered much later how he learned to give such good talks, and it was because he was a Methodist lay preacher, yes. and he used to practice on them every Sunday.
We're talking about statistics and redundancy. But do the ideas of the redundancy and coding, do they relate to what you did in the frog? The other stuff you
Yes.
told me, the notion of noise and statistical variability of signals leads you into these ideas.
Yes, and there was a paper by Atneve, which actually I didn't know about when I first read these things up. Somebody drew my attention to it, and he knew a lot of the psychological phenomena and so on. It seemed that you could get insight into things in coding, like, for example, the adaptation of sensory discharges. The fact that if you put a finger on the skin, it only sends impulses out for a fraction of a second or a few seconds at most, and then it goes quite quiet. And that's because there's no more information. Once the brain's been told of something there, it doesn't need to go on telling it, And in the same way in space, if there's you don't need to respond to the whole of the uniform area, because it points around the edge tell the brain that there's something within there. So that it linked up with, and likewise, with the sensitivity to movement. If only I'd said, and you only have to go on looking a little bit longer, and you'll find things for orientation too. But I didn't see that, unfortunately.
I noticed there's an article here titled "The Brain as a Predictor."
Yes,
now that seems very prescient. Isn't it? It's not about top-down processing.
That was for a magazine. What was it? Grant.
Cambridge Opinion.
Was it Cambridge Opinion? I think that was, yes. That was something that Jonathan Miller edited. That was actually written when I came back. Oh, that must have been sort of early 1950s, is that right?
No, it was 62.
62, okay. 62. That was when I was a teaching fellow at King's. And they asked me to write that, so I said, so I did it.
Was there, when did computer science start? And when did people start wondering whether they could make computers recognise patterns or see things? Did you meet people like that?
When did you see the light with computers?
Well, again, with this, the Ratio Club, Turing, I remember, came there and gave us a talk once. And
Who?
Alan Turing.
Oh, Alan Turing.
And Dennis Gabor, I don't think did, but Donald Mackay was, I suppose, at the beginning of that time, was a PhD with Dennis Gabor, and Philip Woodward was a computer scientist at RRE Malvern. So it was easy to see. And the most advanced equipment people had those days was one of these averaging computers, which of course did add an order of magnitude to any repetitive sensitivity for any repetitive signal.
Did you ever do post-stimulus time histograms? I remember somebody once trying to find the history of the histogram, and it was sort of middle 60s where people started, instead of showing individual pictures of spike trains, started averaging.
Well, yes, we did. The paper with Myung Yoon and Bill Levick on the sensitivity of cat retina to individual quanta. That must be 1970, I should think, isn't it?
Yes, there we go. Yes,
using them,
research.
Yes.
Yes.
That was well into the histogram era.
Yes. So, again, the sort of round about 1960, you're speculating about what the nervous system's actually for.
Yes.
But you still just go on and measure it for, and there's a lot of work about small signals and noise, a lot of detailed work with Bill Levick.
Yeah.
And you said almost as an accident, you also did rabbit retina in a sort of way reminiscent of the way you'd studied the frog.
Well, that was an accident. That was because the I went on sabbatical for a year to Berkeley, planning to do experiments on a cat, and the cat colony that the School of Optometry used. The School of Optometry is next door to the law school, and they had the basement of the law school where they kept their cats, and they had an epidemic of fleas, and the room above the cat colony was where all these secretaries of the high-powered lawyers lived, and they started getting bitten, so they complained to their bosses. So the bosses told the School of Optometry, take your cats away. So we had to. The only thing that was left there were other people working on rabbits, but they weren't kept there. So we had to use rabbits instead. So we hastily had built a head holder for rabbits and found out how to prepare them and so on, and an eye holder, and did the experiments on rabbits instead.
How did you hold their heads? Because we used to do lab classes here, and when you put it, the ear meatus point in a funny direction, so when you put the ear bars in, it just pushes the animal off the apparatus.
I don't know, I can't recall how we dealt with that, but maybe that problem had already been solved by other people doing. Elwin Marg, I think, worked on rabbits, and I think he'd already solved that problem, so we didn't worry us.
Somewhere in this, well, you went to Berkeley for a year.
Yes.
On sabbatical,
yeah,
or some such.
And I came back here.
For not very long?
No, I think it was about a year.
Right,
what happened was when I was Gordon Walls was there when I first went, but he had a heart attack towards the end of the time there, or just after I'd left, I think maybe, and they offered me his job. So I thought,
must have made a good impression.
Yes, probably.
Yes. And did you meet Bill Levick in that year when you came back here?
Yes, he'd come over to work with Fergus.
Right.
But he wanted to do neurophysiology, and there was and Fergus wasn't into neurophysiology.
Fergus Campbell.
And he wasn't into neurophysiology, so he didn't so much offer him to me, but palmed him off on me, and very pleased I was, you know.
So you did lots of rabbits up.
Yes, just
on F floor.
Yes.
Smart. My PhD in that room too. There was a purple stain and a crack.
Yes.
And people said this was where Bill Levick was making a front silvered mirror, and the silver nitrate exploded one night.
Yes, that's right.
They painted it. It's gone, unfortunately.
Can I just ask one thing? When all this research was going on, were you doing any teaching at the same time, or only just supervising PhDs?
I was supervising in King's. Mm. But that was a college lecturer job, but it was, I think, 6 hours a week or something. It wasn't exactly the morning.
So did you lecture in the Part 2 course?
I did. Yes. I also did Part 2 lectures. Yes.
Well, on vision.
Yes.
But you didn't teach in preclinical, or did
you? No.
When you went to Berkeley, and then you had to set up a lab, or you had to find the old rabbit colony again.
Yeah.
Did Bill Levick go with you?
Yes.
Yes. So I thought you had worked longer than just 1 year.
Yes, we did quite a lot of the motion experiments in that lab there. Yes. But in fact, I think we did most of them there. And then when we got to Berkeley, we started working on the cat.
Yes.
And trying to do the experiments that I'd planned originally to do at Berkeley. But they didn't work out quite the way we expected. It took us a long time to sort of get going properly on that.
Was the result ultimately different, or was it harder to get the result?
Well, we came across quite a lot of other interesting things in the course of it. I mean, the actual way that the retina sort of normalizes for a mean luminance, and perhaps also for the variance, the standard deviation of fluctuations as well, I think I have the impression that we were a bit slow in the uptake as to what that was all doing, you know, and and we got a lot of interesting results about quantum spike ratio and things like that, and how things do shift around. But I don't think we ever made a very good story of it, which I could do better now. But
was William Rushton sort of coming back into this? Was he about the same time?
He went over to Berkeley to work with Gerald Westheimer.
Oh, and then did he also go to Evanston, to Christine Renroth? Or
no, he went to Florida, and with Matt Alpin too, didn't he? He worked with Matt Alpin, but Matt never went to Florida, I don't think. I'm trying to think who was at Florida with him, whom he only spent several years, most of the time in Florida, didn't he?
Yeah. There was this debate that I very distinctly remember about adaptation pools and
yes.
how large an area you got adaptation over. Was it a receptive field or a rod? And I don't know, which side were you on at the time, do you think?
Well, I was trying to explain things in terms of dark light, of the idea that the rods were generating pseudo-quanta, so to speak. They were generating a response that looked as if the rod was absorbing quanta, when it actually wasn't. It was regenerating rhodopsin or something. So if the regeneration of rhodopsin made it think it was absorbed a quantum or something like that. In which case, if that had been the case, you didn't need the mechanism that William had postulated. I think what finally turned out to be the case was that we were both wrong.
What was the origin of the extrapolation down to one quantum?
Well, that was simply the calculated number of quanta absorbed from the flashes of light we were putting in, and then plotting histograms and finding a number of impulses from a single neuron was more than the number of quanta that had been absorbed.
So is that still accepted?
I think so, yes.
Because William Rushton had this notion of adaptation pools and networks and things, but I've never actually seen one drawn out to quite know over what area it would happen. And
well, I have grave mistrust of those experiments that he did with stabilized images, because of the trouble we had, and it's very difficult to know whether the image is sharply focused. And
this was the experiment of, in theory, flashing a very a bright dark, a square wave pattern, and then showing that the threshold in the darker bit has been affected as well as in the brighter bit.
Yeah,
that was
what he would predict.
Yes,
but the trouble is, so would we, because the masking effect of the noise from the strongly illuminated bars could easily interfere with the detection of light in the
inhibition.
Well, no, not necessarily by inhibition, but by adding noise to the signal. If the system couldn't separate out between the bars,
because there will be convergence over
then the noise from the bars would hinder the perception of light in the between the bars.
So you've got 3 factors limiting reliable detection.
Yes.
What were the 3 factors? Because there was a paper a decade earlier saying something about 3 points about lateral inhibition, but maybe that's different. I'm sure it is. 3 factors limiting reliable detection.
No, that was different, yes. I don't know what the other 3 factors were. 3 points. The 3 factors were loss of absorption, bleaching, noise, and I can't remember what the other one would be.
I think it was one of the first papers I ever read, actually.
Is it James
right.
Delano?
Yes,
Shall I go and get it?
1969 it was.
Oh,
I seem to remember it was on one of those, that strange pink paper.
Is it volume 200, page 1?
Yes, sort of millennial.
Yes,
and
yes.
it was sitting in the reading room when I was starting my PhD, and I avidly read anything, and that was one of the first things that was there. And I think I could do 2 of the factors, but the third one I found rather difficult.
I must look that up.
There's very interesting things, so that changes in the maintained discharge with adaptation level in the cat retina in 69 with Bill Levick is very similar sort of idea to what you did with Fitzhugh and Kuffler, wasn't it? Maintained activity in the cat's retina in light and darkness. Did you come back to it with another idea?
Well, I think that the new, I think it's in that paper, isn't it? The fact that there are cells which behave regularly.
Yes. Were you recording from retina or from the nerve with Bill Levick?
From the retina.
You really were?
Yeah, yes. And those, as we now know, are the rhodopsin-containing ganglion cells. It's not
melanopsin.
Melanopsin-containing ganglion cells.
Right, so you were finding them?
Yes.
Because there was this very interesting thing that sort of happened in the late 60s, early 70s, where people began to realise, unfortunately, that the experimental technology you used actually seriously affected what cells you recorded from.
Yeah.
So I hadn't quite appreciated that you were one of the original finders of W cells. Of course, Bill Levick then went on to do some very seminal work with Brian Cleland, and then Heinz Vesely, wasn't it?
Yes.
Yes, some extraordinary work in the late 70s.
Yeah.
So his direction will, perhaps have started there to find some sort of slightly odd ganglion cells that people weren't finding.
The main point, or I think that that's so long since I've looked at those papers, I've really forgotten what's in them.
The work on the Perginy shift, was that a digression, or was that something that you worked
No,
with?
that was the fact that in all animals that have a Purkinje shift, let's say when the wavelength changes as you go to lower luminances, it always goes to shorter wavelengths.
Cyril Rasher.
And
Richard Adrian.
I hadn't appreciated until
Ainsley,
that time that the spontaneous decomposition rate of a photopigment becomes much less as the activation energy becomes less, which is accompanied by the shift of the. So the pigments that are only sensitive to blue light have a lower thermal degradation rate. So that
a lot of papers in here I read.
That seemed to fit in with the fact that high sensitive pigments are shorter wavelength.
Yes.
That's all just been wrapped up actually by King-Wai Yao. He's got all that.
One of the first research I did was with Geoffrey Arden.
Oh right.
On the tapetum of the bush baby.
Oh wow.
It had a riboflavin tapetum, looks like gold leaf.
Oh, right.
And the idea is that the light comes in the centre of the rod, of the rod maximum, and then it comes at a higher, a bluer wavelength than the rod maximum, and that shifts it into the green, because it fluoresces and pushes it back through the.
Oh, does it actually fluoresce?
That was the hypothesis.
Yeah.
I'm not sure we got a proper. It was all done with electroretinography.
Yeah.
With Dartnell.
But I don't think that riboflavin
actually fluoresces.
Yeah.
But
so it would shift the peak wavelengths, but it would do it by absorbing light, not by creating light. So that doesn't work out.
But that was at the beginning of my career.
I've got your 3 factors. Thank you, Anne. the quantum to spike ratio, which is the mean number of additional quantal absorptions required to produce an additional impulse, presumably on average.
Yes.
The temporal course of the response, which determines the time interval within which the maintained discharge is modified, and the statistical distribution of the number of impulses that occur in this time interval in the absence of the stimulus. Did you do gamma distributions in this paper?
Yes.
I vaguely sort of one of the great mysteries for me was maths, and I think there were gamma distributions.
Yes.
Didn't you tell me your brother once wrote a table of gamma distributions?
No, that's not my brother.
Oh.
There was Barlow's tables may well have had gamma distributions. I don't know.
So,
that's interesting.
Yeah. And I was sort of just thinking that you did worms, frogs, Ommatidia, we missed that, Ommatidia,
right, yeah.
Cats, people, pigeons, rabbits, any other animals? Pigeons. How did you write a paper on the pectin of the pigeon's eye?
Yes, that was... what was it? Who's that with? What's his name?
Oswald.
That's right.
T. Oswald.
He did a project with me, and the idea of that was that the retina is concave, so that if the image of the sun is at one part, there's a very, very bright spot there on the retina, and it's illuminating all the rest of the retina, because it's not as if it was flat or curved the other way. This can actually see that spot, so it will be flooding it with light, and that would have a very big effect in masking effect. And in fact, masking effect, there's a lot of interest in this in connection with car headlights and things.
Is it not truly concave, or is it another function?
It's not necessarily spherical,
So
but
it's a focus.
No, but it is concave,
Yeah.
so that the light from any point, which is another point, and of course, if it was 180 degrees away, it'd be worse. But because it's oblique, but it's still quite a lot light gets there.
And the scatter.
Yeah, and that will be enough to raise the threshold very considerably. And the idea was that by having this structure which sticks out, pointing towards the lens, you will intercept that light, so that if the sun's image is one side, the whole of this, the other side of the pectin, will be in shade. Yes, so it's an intraocular eye shape. And I think that's right. I think that's one of its functions. It also has another important function of providing oxygen it's how the vitreous is oxygenated, so to speak. So a lot of the receptors get their oxygen from there. And I wouldn't like to get into a fight with anyone as to which aspect had greater survival value, but I think the one we pointed to there is genuine.
The various things that you started sort of between sort of 47 and 1970. Were there ones which, looking back, you're disappointed that either you or maybe other people haven't been able to pursue or didn't want to pursue? Or
I think people ought to pay more attention to the absolute performance of these statistical tasks, because the world is full of people who run down what the brain can do and point out how illogical people are and all that. But in fact, when the brain wants to do a good statistical job, it can do it very well. You have to choose the right job. I mean, it can't do every job very well. And there's nothing like an argument from statistical efficiency for knocking down theories. If a particular theorem says that you're going to need 250 quanta to do a job, and you show that the brain does it with 10, then that theory is in the wastebasket. And there's no other way you can knock things out as conclusively as that. that I know of. So I think that it's people ought to pay more attention to the optimum performance. Is it?
Most of yours, yes. But were there any things you hinted that maybe your work on dark adaptation pools, and maybe you would have interpreted it differently now, but or can you think of, with all these modern techniques like reverse correlations and confocal microscopy and calcium imaging, whether anything you did would have been done better or quicker? Or
with regard to doing better, I mean, I'm certain that if I submitted papers at random from that list, at least 50% would be rejected now. Because the standards just are much higher.
But it's the ideas, it's the ideas that are important.
Well, yes, but it's very difficult to publish an idea without any facts at all supporting it. It's called mere speculation.
I mean, these are the groundwork. The people who work now look at these to, building on them. They're not doing the thing from the start.
I don't know, perhaps what Horace is saying is that nowadays it's quite difficult to send in a lot of bricks and build them up. They somehow feel it has to be a completed project, like you still don't know how the brain works. So
well,
we
it's
can't
not that, but the
publish
actual technical standards. I mean, you pointed out that I might not have got ethical approval for putting a person's head on a concrete block and wedging it with things and so on.
Well,
putting mercury in it.
But it's, of course, what you do to animals matters.
Yes.
Yeah. Tim Bliss, one copy of his PhD thesis is How the Brain Works, the title.
Your experimental work, apart from, is it 1 or 2 digressions, the one on binocular disparity, it's almost entirely about the retina.
Yeah.
Yes, obviously that's where a vision starts, and if it doesn't leave the retina, you won't see it, I think.
Yes.
You said to me yesterday that Lord Adrian somehow didn't really believe that you would be able to study individual brain neurons and understand how perception or behaviour worked. It was just too difficult, and it would be a network.
Yes, well, that was the generally held view was, it was a very complicated network, and even if you could record from a single neuron in the brain, you wouldn't be able to find out where it fitted in, which is perfectly reasonable if you believe that that everything in a brain depends upon millions of neurons being active. You're not going to get very much from a single one. But they were wrong. The part played by the neurons is bigger than that, and big enough so that you can see, as you go from stage to stage, significant changes happening in a direction which makes some sense.
Yes, your experimental work is mostly about the retina, but the speculations about what it's all for and the neuron doctrine, the perception paper that is their complete impact factor, I've been told. These are not just about the retina, are they? They're sort of grand.
No, there are more grand ideas, yes. I was talking about redundancy reduction, the importance of redundancy in vision, starting actually before any of the first things were published, because it was a talk I mentioned at the journal club. But it wasn't a journal club, it was a discussion meeting I went to, that was organised by Zangwill and Thorpe. And I gave a paper there about redundancy, importance of redundancy, which I think was in 1956 or 57. So I've been talking about that for quite a long time, but it's only quite recently that this has been taken up, and other people have understood what I was saying.
Did you coin the concept or the terminology of redundancy?
No, that's Shannon. Shannon's redundancy plays an important part in Shannon's theory of communication. And the engineers were always understood immediately, because if you understand about redundancy, you can send 10 messages where one grew before, if you see what I mean, to mix metaphors. So they understood that correct immediately and applied it. I mean, that's why, the web's so cheap and all that, because it has been understood, and you can understand how to pack information send information down wires much better. But to some extent, I and other people at that time were misled by thinking that it was only important in getting a more Shannon information per functional unit. So hence I talked, and other people did, about redundancy reduction. But that's not really what's at stake. What is at stake is that in the ordinary way we find our way about in the world, if we're dealing with an extremely complex world where everything is interacting with everything else, and in such a world it's extremely difficult to learn what is truly associated with something else, because it's all meshed up and things are correlated stronger. So you've got to understand the statistical relationships in the external world. Now, it's true that understanding those statistical relationships is what enables engineers to make proper use of optical fibers and things like that, and that's the importance of redundancy for them. But for the brain, it's devising a representation on which you can learn. You see, when from Pavlov and every other learning theorist always assumes that different stimuli they use are independent of each other, and of course they're not. Or you have to work hard to make sure they are. And the messages that come into the brain aren't like that. You've got to do something to make sure they're decorrelated. They're not lurking in that information, a whole mass of interdependencies which are getting combining, because you're going to think something's association with food is an independent fact, whereas in fact that may be strongly dependent on something that has already been taken account of and discounted.
So what level? We're not talking about lateral inhibition.
Well, lateral inhibition is a step in that direction.
Yeah. Or mutual inhibition.
But for example, if you look at Jeff Hinton has got an article in recent Proc, I think, about learning vision, I think it's called. And in his system, almost the first step that he talks about at all, it's not to do with light and dark adaptation or anything like that, but it is to do with getting a covariance matrix, in which the values are low, getting rid of covariances. And that's just the kind of thing which I think is the right model for what goes on in the cortex.
So these are general sensory things, they apply to tactile sense, auditory.
Yes, but according to that, it should be applied to any other source of information. So you've got to make sure your tactile information is not simply copying what you're getting by visual information, for example. If so, then paying attention to the tactile information won't buy you anything at all, because you've already taken all the information that's there is also in the visual one.
Well, then you have selective attention.
Oh, well, yes, that's attention comes in then.
You have an article here called "Is Neurobiology Totally Non-Mathematical?" in 1976 in Lectures on Mathematics in the Life Sciences, Volume 8.
Yeah.
Was neurobiology totally non-mathematical? And is it still?
Well, there's a lot of misapplied mathematics in the theories about networks and so on and so forth. Partly because of just what we've been talking about, that it is not always appreciated just how interdependent all our sensory messages are. But I can't remember what I said in that paper.
Yeah. I wonder, do you think you really did talk about mathematics, sort of, I don't know, Bessel functions and von Euler things, or it's sort of in the days before computational studies? And it seems to me a lot of the ideas that you suggested about redundancy reduction and decorrelation have only really begun to look credible. That's not quite the right word, is it? To see what they really mean when you can do a computational analysis of how you believe a vast number of neurons would respond. So it actually means computer modelling, if not mathematics.
Well, I do think that's true. I mean, in the old days, one could, utter.
You waved your
make
hands.
theories and waved your hands.
Yes, I think you.
And
Classical ones.
Some people were convinced, not all, but some.
Some were very good hand wavers.
Yes, you waved your hands and said, if this neuron inhibits that one, and this neuron inhibits that one, then this will sharpen that one's tuning curve.
Yeah.
And it sounded great until
somebody
you
said,
actually tried to write a computer program that did it, and it wouldn't
you
work.
find it doesn't tell you anything of the sort, yes.
Where do you think the ideas of, or how would you pursue the study of coding and statistical efficiency and so on? How would you pursue it now?
Well, I am pursuing it.
Yes,
or trying to.
Maybe experimentally, perhaps. What sort of?
Well, I think that what we're doing now, is trying to do psychophysical type experiments on the operations that it looks as if in the light of all these ideas about signal noise redundancy and so on, is likely to be what the early stages of visual processing in the cortex are actually trying to do. So we're using the ability of the eye to detect faint streaks against a background of noisy dots. And it turns out this is a task that the eye can do quite well. We haven't got the final figures for the absolute efficiency yet, but they're going to be of the order of 20 or 30% or something. In other words, the system is not a complete idiot at this. It's doing really pretty well. One of the things that you can compare with this is how well you can see streaks which are generated by changing the mean density of the dots. And if it's in a streak, then you detect that. You signal that it's going to the left, not the right, and so on and so forth. And you can put that into the statistical calculations and show that it's making good use of all the information in those dots.
These are psychophysical experiments.
Yes. But you can also produce streaks by a different manoeuvre, which is the glass patterns, where you have you've probably seen them at some stage in your life, where you have somebody puts up an array of dots which look like a spiral, but in fact they're only made of pairs of dots, no more than a pairwise dependency, and yet you see a complete pattern of streaks and so on. You produce dots like that, and not only is that also done with a high efficiency, but it's done at the same time as the others. I mean, you can do the same experiment, and you get the same result with regard to efficiency, and yet the mechanism required to do it is totally different. So there's another kind of analysis there leading to the vision of streaks, which is autocorrelation.
These pseudo-movement effects.
Yes, that's right, pseudo-movement effects, but there isn't any actual movement there.
Is there an experiment that you can't quite do now that you'd love to do? I mean, that's if you had.
Yes, but some of them can be done if only I can find somebody who can. Who's got really full control of the MATLAB. I know one person, but unfortunately he's in America.
But this is why you're using David Berry, who's a physicist, really.
Yes.
Yeah.
Yeah.
If you had a nice clean lab and legal permission and you had frog retina again, is there an experiment you'd do on the frog retina, or would you do something differently? And you had these clever electrodes that can record from lots of things at once and convince you that they're able to separate the signals.
Yes. I haven't been directing my thoughts in that direction. I think that the frog's brain is simpler than ours, and I've progressed from the very simplest physiological mechanisms. Nothing can be simpler than the absorption of a photon in a receptor, to things which are much more complicated, and these are the ones that now...
It could be simpler, they might not have adapted or anything.
Yes.
Yeah. I've just sort of caught a glimpse here of a very important 1976 paper called The Language of the Brain, or The Languages of the Brain, but I guess that's important for other reasons.
But you haven't yet waxed philosophical and religious like Eccles.
No.
Or Sherrington.
Or Sherrington.
Well, the nearest I got ideas about consciousness, which is, so to speak, something I learned at my mother's knee, to bring us back to the beginning again. You're not going to understand anything about consciousness until you know what its survival value is. And to many people, including William James, by the way, which rather surprises me, he couldn't think what kind of survival value consciousness could possibly have had. And that's doubly surprising, first of all, because he could think about a lot of things, and second is he's an Irishman, and Irishmen are notorious for the way they can wave their hands and talk, and I maintain that we couldn't communicate with each other if we couldn't also talk to ourselves. And that is what consciousness is. We're describing what's going on to ourselves, what's going on in our own brain. And if we can't describe it to ourselves, we can't describe it to other people. So that if we didn't have consciousness, we couldn't live the social life we do. We couldn't have this kind of discussion.
Well, what's the alternative to consciousness apart from unconsciousness?
Well, I gave a talk about this once, and to my horror, at the end of it, somebody got up in the audience and said, "I'm sure you know Nietzsche's soliloquy number 293 or something." And
sounds like Dennis Noble.
It wasn't actually. It might have been. And I didn't know it, so she told me what it was. And he had said that if it wasn't for consciousness, we would be like a solitary beast in the jungle, that doesn't communicate with its conspecifics, leads a solitary life, and that manages to get by all right in a few social activities it does engage in, probably not for public viewing.
But it's conscious.
Are you sure?
Well, of course not. I don't know. We don't know what's in other people's heads. But
I don't think it's conscious. I don't think it's capable of communicating, describing the internal contents of its brain to another tiger.
You've got something called Nature's Joke, a conjecture on the biological role of consciousness.
Yes, that was one.
Nature's Joke.
That was it was pointing out that consciousness is not what we think it is, and that we think it's concerned with the internal working of our brain. So you have people talking about possible consciousness of a brain in a dish. I don't think that's possible at all. I think that the conscious part of our brain is what enables us to be the kind of social animals we are, and that we won't understand much more about its nature until we understand in more detail how it provokes the survival of the human race, which is, when you come to the more and more you think about it, the more and more important it becomes. I mean, even the baby has to cry to get fed and so on.
Well, you said you allow us to be the social animals that we are. So, in case of, say, severe autism, would you think there was some difference in consciousness?
Yes, I think I would. Yes. The trouble is, when you start saying things like this, you're liable to be accused of thinking that this entitles you to treat an autistic child like an animal, or something like that, or an animal, even like an animal that you don't think does have consciousness, which of course doesn't follow.
No.
I mean, it's. If I say that I think that autistic children are not conscious, they will say people are liable to jump to the conclusion that I think you can use them to make shoe leather or whatever it is, which doesn't follow. I think there's an interference with consciousness, should we say.
Maybe we should lighten the discussion.
Oh, I think that's pretty light-hearted.
What are the plans? What have you got? You're working on this psychophysics, which you're, in a sense, trying to find out what the visual cortex is really, really good at.
Well, yes. Well, there are now two things. There's the cross-correlation, which is what you see gratings and things with, and there's autocorrelation, which is a much more interesting operation because it's not, and it's totally different. Though they both depend upon correlation, the multiplication of two arrays, I mean that part is the same, and something differences. But the arrays are totally different. One is a fixed array in the brain and a variable array in the image, and the other is what you're finding that these two arrays in the image are the same as each other if you do something to one of them. So if I translate this like that, it becomes the same as this, and if this happens in time, then you see motion. So it's a totally different operation, and it's I don't understand it at all. I mean, I don't understand its neurophysiology at all because I think it's what goes on in these pinwheels, but that's a very highly localized operation, and in many of the forms of symmetry which are clearly important in the brain, it involves interactions over a very big distance. For example, I can see that this hand up here is, by means of autocorrelation, I can find that those two images are the same. All you have to do is to flip one over and put it over there, and they become the same. So that it's the same basic operation as the autocorrelation, which I think we now know goes on in primary visual cortex. But how that is represented neurally, I don't understand at all.
Have you been interested in stereopsis? Yes.
Yes, I've been interested in that too. Yes, because there's motion and there's glass figures, which is where the simple form of autocorrelation. There's motion, which is slightly more complicated because it involves a time delay, and there's stereopsis. But there's also rhyming in poetry and the detecting the rhythms in music and things like this, which are best done by autocorrelation.
Yeah.
And presumably there are
binaural hearing.
Yeah, and binaural things. Yes.
Do you think if you were doing microelectrode work and you record from individual neurons in the retina, or perhaps more interestingly in the brain, and you characterize what they respond to and so on, and then let your animal or your person go about their everyday business and do things for 6 months and come back, if you were able to record from the same cells, would you get the same behaviour, or do you think they would have changed?
Sounds like a viper.
I think it's a very interesting question. If you can, what one really would like to be able to look at a Golgi preparation, so to speak, after a few months of a particular pyramidal cell, before and after 3 months of ordinary use, and see whether detail for detail, they were saying. My guess is that probably not. Was much more active.
You have these ideas about adaptation and learning and sort of learning to see the improbable and so on. That maybe the system is a lot more dynamic than would seem to be the case if you just spend sort of a few hours looking at a bit of it and
I think
then go away again.
I think a lot of it does depend upon maintaining dynamic equilibrium.
Yes. Yes, I guess the world's sort of moderately constant, but maybe what's redundant today might not be quite as redundant on another day.
Yeah, you haven't got to assume the world's constant, that's true.
Yes. I was trying to think of who the averaging computer, that was George Dawson, of course.
Oh, yes,
yeah.
Yes. Yeah.
With capacitors.
When you finish the psychophysics, what are you going to do next? Have you got
I've just been
thought?
Just been trying to write a paper, and I think the time will come which I shall always, when I just finished a paper, say, well, I'm not going to write another one. But it usually so far it's been the case that I have written another one, but it's not going to go on forever.
It's 199, it says.
Yeah. Yes, I'll do one more.
Yeah. Are you still talking and lecturing? I mean,
I don't actually do any teaching.
No, I mean informal.
Yes,
or
yes.
informal talks.
I was thinking, yes, a lecture or two.
You've also had to do Darwin this year, haven't you?
Yes, I have. I managed to get away with only one talk, one after dinner talk about what it was like being a Darwin.
Yeah. You claim you knew him.
Hmm?
Do you claim you knew him? The students probably think you did.
Were you brought up in Cambridge?
No, but my maternal grandmother lived here.
Was she the one who wrote the book?
No, that's Gwen Ravrau. She was she and my mother and Francis Cornford sort of grew up together. Mm-hmm. So I saw a lot of the Cornfords too.
You didn't speak of your father. What?
He was a civil servant and not. He was very keenly interested in the origin of words. We always used to have arguments about meaning of words. So there was the large version of the Oxford Dictionary was always in the dining room, usually consulted at some stage in a meal.
So he worked up in
London.
In London.
Yeah.
So he commuted.
Yeah.
And what about the Cambridge Instrument Company?
That was my paternal grandfather. That was Horace Barlow, who was mayor of Cambridge. And he was Cambridge's equivalent, so to speak, of Bill Gates or somebody. But that was before people like that made as much money as they do now, unfortunately. And he was, I think he was not considered, it wasn't considered that he'd really behaved as a Darwin should behave, even though, of course, because he started a company and things like that. He wasn't. But of course, I mean, the Wedgwoods were not only was Emma Charles's mother was Wedgwood, and countless other cousin marriages before and after that. But they were very much the same tribe.
Was Charles Darwin a Quaker?
No, the Wedgwoods were. And I think they were.
Interesting how many of those sort of 18th, 19th century big industrialists were all Quakers.
Quite
like
a lot were, yes.
People, they built little towns to keep their workers in.
That's right.
Yes.
Keeping on the straight and narrow.
Yes.
Glenda Huxley, that was a Quaker family.
Well, that was
Pease.
Chenda Huxley.
Yes.
She was a Pease.
Yes.
And that was a proper Quaker family.
The Finns.
Pease.
Pease.
Yeah.
So is your family still, are you all in, is there a big group in touch, or have you all gone your separate ways?
Well, every now and then we meet, usually at a funeral or a wedding, occasionally a wedding, something like that. Yes, I think we probably see more of each other than most cousins do. It's quite a large cousinhood.
That's the Barlow side.
No, well, I keep in touch with one or two of the Barlow side cousins too, but most of them are on my mother's side, Darwin side. And last year we saw nothing but each other practically.
Yeah.
And this the Ruth, the poet.
Yes.
Now where she's
my niece. She's my sister's daughter.
Yes. So she's Darwin from.
Yeah.
Yes.
Mm. So I think we've finished.
Can I just ask you? What about your own children? Are they sort of Darwin-minded or?
They get on quite well with their cousins. Actually, they like some of them, but I don't know how many generations go on being on speaking terms.
And what has anyone taken to science of your lot?
Of my particular lot, one of my daughters is a mathematician, but and she has a construction named after her.
Oh.
Something to do with algebraic geometry or something. I don't understand it at all. But she had children and found she couldn't give lectures and have children at the same time, so she's given that up. One of my current lot is one of the very few people who I welcome in my lab, because she's got all that it takes to be a good scientist. You know, she's very observant and very good with her hands, and has a lot of common sense, and seems to be able to get equipment to work, and even to repair it at times. But she's at Oxford now, and she shows no signs of devoting her talents to science. She's running the Pembroke College Boat Club at the moment.
Is there religion in the family?
I'm afraid there are pockets of religion.
No, I think I said the same.
Well, we thank you.
Yes, well, we're very pleased and it's very interesting. We learned a lot, and I'm sure other people will enjoy hearing it when it's all transcribed.
Well, I feel sorry for the person who has to transcribe it.